Good morning, good afternoon, good evening, everyone. In case you missed yesterday, a brief introduction. My name is Wang. I'm from the Office of Research and Standards, Office of Generic Drugs, FDA. I hope everyone found the presentations and discussion in yesterday's sessions informative and helpful. Before we start a new round of exciting topics, I'll use just a few minutes to give a quick recap of the in-person discussion sessions we had yesterday, mostly to serve our online audience, hoping many of you will be able to join our future workshops in person. Just a brief description, we have a big room here and we had 10 tables in the center of the conference room, and each table we had roughly 8 to 10 people, including FDA, industrial, and academia. The first half of our discussion, we primarily focused on characterization of complex excipients and determination of qualitative Q1 sameness. One common recognition among all participants is characterization of polymers is challenging due to various reasons, such as lack of reference standard polymers, inherent variability of certain polymer characteristics that could be from batch to batch from the same supplier, or when you compare the same polymer from different suppliers. And also for some analytical methods, the data could also be variable depending on the nature of the method. Almost all tables discuss the PLGA polymers, of course, because that's one of the excipient that is used in many long-acting products. The other example polymers considered challenging examples included EVA, silicon elastomer, polycarboxyl, and CMC sodium. Several tables also mentioned the impact of polymer grade. Questions include whether different grade is considered Q1 different. How may an applicant justify the difference in polymer grade to be able to file an ANDA when they are aware the potential difference? But many times, the information on the polymer grade for the reference product is not readily available, and reverse engineering to precisely identify the exact grade can be challenging due to overlap in certain polymer properties. In addition to more scientific understanding on the impact of those differences in grade, and then develop appropriate analytical methods. One comment was it also would be helpful if FDA can improve the clarity of the information on product description section in the labeling of the reference product. And I believe a similar comment was made in the online chat box yesterday during the session. So second half of the discussion covered multiple topics, including IVRT methods and product-specific guidances. One commonly seen challenge during IVRT method validation is how to make altered formulations with meaningful changes to demonstrate discriminatory ability of the method. What are considered meaningful changes? What if a method cannot discriminate an identified critical quality attribute? With regard to selection of apparatus, how to decide if one should go with compendial methods versus develop a novel methods, and when to use that for QC versus for bioequivalence purpose. All those are considerations to be included during that decision. And is it required to always use compendial apparatus to establish similarity in risk profiles in addition to the most referred standard preferred or use the statistic F2? Well, what are other possible statistical methods? Because F2 may not be the most appropriate for certain dosage forms. Then for complex generic products, in many cases, IVRT does not only serve the purpose for quality control, right? It also plays a pivotal role in bioequivalence determination. So more specific recommendations on how to conduct IVRT to support BE as a one-time study in the product-specific guidance could be beneficial. Similarly, more specific information on recommended studies could be helpful in the PSG So one example is to further clarify for a product with multiple strengths, whether testing each strength in vitro is necessary, or if the strengths only differ in field volume with identical composition, then maybe test the one or sometimes highest, lowest strength as a bracketing strategy could be sufficient. In addition, it is recognized that more recent PSGs have already expanded the scope to include information beyond just bioequivalent studies, which is recognized as helpful, and more information beyond just bioequivalent studies could be considered, and that will further facilitate the generic development. And another comment is when we recommend comparative characterization studies, if appropriate, more descriptive information on how to establish comparability is beneficial. So this is just a brief summary of the main discussion points. I hope online audience feel these discussion points are also relevant to your experiences. Due to the time limit, we wasn't able to take many of your questions from the online audience, but please continue to submit your questions and comments to the chat box throughout today's sessions. We are collecting all of them, and we will try to address them through our future talks, guidances, publications. With that, I'll pass the floor to our moderator of the next session, Dr. Yogeeta Narkar. Thank you. Thank you very much. Hope you enjoy the second day.
Greetings, everyone. Welcome to session 4. I'm your moderator today for this session. My name is Yogeeta Narkar. I'm from Office of Lifecycle Drug Products within OPQ FDA. Today's session will focus on scientific and regulatory considerations when developing product-specific guidance and assessing ANDAs. So our first speaker is Cedar Boakye. She's a senior staff fellow with OPQ FDA. So please welcome Cedar.
Hello, good morning, and welcome. Thank you very much, Yogeeta. In my presentation today, I'll share with you some of the agency's perspectives and expectations when it comes to quality assessment of polymer-based vaginal systems and implants. In the Office of Pharmaceutical Quality, we believe that everyone deserves confidence in their next dose of medicine, and that pharmaceutical quality assures the availability, safety, and efficacy of every dose. Please note that this presentation reflects the views of the author and should not be construed to represent the views of the FDA. My outline basically will cover giving a very brief background to what vaginal systems and implants are, and share with you our current expectations when it comes to their quality assessment, and share with you some of the quality issues we have observed over the years during our quality assessments. You will all agree with me that vaginal systems have gained a lot of attention currently, even though they were developed many years ago, because of their versatility when it comes to their ability to achieve extended release over weeks to months. Also, because the vaginal mucosa tends to be highly vascularized and permeable, it tends to be an ideal route of administration for a wide range of drug targets, for targeted delivery, either for local action or for systemic effects. And vaginal systems tend to be fairly easy to administer, hence it improves patient compliance. There are many different designs of vaginal systems currently under research and development. However, vaginal systems that have been assessed by the agency and that have been approved fall under one of three designs. The first being vaginal the matrix vaginal systems, whereby we have the drug constituent dissolved or dispersed homogeneously within the polymer matrix. And an example being the mucosa. For the reservoir vaginal system, we have the drug constituent dissolved or dispersed within the polymetric matrix core, which is then surrounded by a drug-free membrane or polymer. And I've given examples here, including the S-ring and the NuvaRing. And the last example of a design that has been approved is the multi-core vaginal system, whereby we have the drug constituent dissolved or dispersed within rods or tablets that are again inserted within channels within the drug-free polymer membrane. Because vaginal systems are drug device combination products, there are two aspects when it comes to their quality assessment. The first aspect focuses on the polymer-based materials and related components for drug delivery, and excuse me, and focuses on the engineering and biocompatibility of the device components. The second aspect of quality assessment focuses on the drug components, such as the drug substance and inactive ingredients, where we focus on the critical quality attributes such as the drug release, the drug substance impurities, the drug substance identity, and then assay, as well as microbiology and container closure systems. As is the case of most single entity drug device combination products, vaginal systems, the polymer materials that are used may be considered as both components of the device as well as of the drug components. The primary mode of action for vaginal system is drug. Hence, per regulation, Cedar takes lead when it comes to quality assessment. And then we have the CDRH office providing consult and assessment of the engineering and design aspects. Vaginal systems per regulation are not required to be Q1, Q2 as the RLD. However, it's worth noting that it has significant implication for establishing BE. Hence, Q1, Q2 sameness typically will be a critical assessment issue. It's also worth noting that the API characteristics may impact the API dispersibility within the polymer during blend, as well as on the uniformity of those of the final drug products. Hence, in our assessment, we tend to recommend strongly that applicants consider characteristics of the API, such as the solubility, the glass transition temperature, compatibility with the polymer, the particle size distribution, as well as stability of the API during the shelf life and during the manufacturing process as well. And I can also stress enough, because of the presentations we've had from yesterday, that the polymer chemistry, physical characteristics, as well as rheological properties may have significant impacts on the mechanical properties of the final drug product, as well as the appearance and the drug release. Currently, there are two main types of elastomers or polymers that are used for vaginal system. The first being the silicone elastomers. These are formed through chemical cross-linking of functionalized linear PDMS. And I've provided here examples of vaginal system using silicone elastomers. They include S-ring, femring, and anoreva. The second type of polymers include the thermoplastic elastomers, which include the ethylene vinyl acetate, which is used in NuvaRing. And the third is the polyurethanes formed by the polymerization reaction between diisocyanates. We currently do not have any vaginal systems approved using this polymer. As I've stressed previously, it is really critical to know that the polymer characteristics have a lot of influence or impacts on the mechanical properties of the final drug product. Such as for silicone elastomer, it's worth noting that the curing process, whether it's by the condensation curing, addition curing, or free radical curing, may significantly impact the API solubility within the polymer, as well as may impact the presence of residual monomers or even byproducts or certain impurities, as well as may result in some reaction occurring between the API and the polymer. When it comes to the EVA polymer, it's worth noting that, as we noted from yesterday's talks, that the vinyl acetate content and the molecular weight significantly impacts the mechanical properties of the final vaginal systems, as well as the drug release. Hence, we keep on stressing that applicants consider I've provided here certain factors that applicants should consider when selecting their polymers for use. The first is we recommend that applicants demonstrate strong understanding of the properties of the polymer raw materials when it comes to the polydispersity, particle size, and rheology. And we also recommend applicants demonstrate sense of understanding for the manufacturing process of the polymer as well as the final vaginal system when it comes to cross-linking or polymerization, the type of curing process that is used, and its impact on certain impurities that may be present, specifically extractables and leachables, the impurities and byproducts, residual monomers. It's also significant and highly recommend that applicants consider the drug solubility and physical stability within the polymer course shelf life. When we talk about stability, talking about the propensity of it recrystallizing out or crystal ripening happening during the formulation, manufacturing process, or over the shelf life of the drug product. Again, we expect that applicants demonstrate understanding when it comes to potential of some form of bonding occurring between the API and the polymer. And lastly, we recommend that applicants also demonstrate understanding and provide quality control when it comes to how the polymer may affect biocompatibility regarding maybe resulting in certain irritation of the vaginal mucosa. When it comes to testing of the mechanical properties of the vaginal systems, we can't stress enough that the assessment should be performed on the final vaginal system to support evaluation of the device. And I've provided here some of the typical testing we recommend that applicants consider when trying to demonstrate comparability or Q1, Q2 sameness as the RLD. Some of the tests are provided here include hardness tests, the tensile strength, compression strength, twisting during compression, seal integrity, elongation at break, and dimensions. These are, should I say, standard or basic tests that we would expect to see an application that's submitted for review. I'll switch here and then go into specific other aspects of of our quality assessments because of the numerous questions we receive when it comes to these areas. When it comes to the batches that are manufactured for submission, we want to stress that 3 registration batches submitted should be manufactured with at least 3 distinct laws of the API and 3 distinct laws of the polymer. The registration batch, regardless of the number of units submitted, the batch size, the equipment, and the manufacturing process control strategy should be representative of the commercial production. And in cases where this is not so and there's likelihood of there being a scale-up, we recommend that applicants submit a post-approval supplement. And lastly, for packaging of the final vaginal systems, we stress on the fact that applicants use representative samples from all the 3 batches packaged in the proposed marketing representations as per regulations. And when it comes to drug release considerations, we recommend that applicants strongly demonstrate a sense of understanding of the state of the API. Is it dissolved? Is it existing in a solid state dispersed throughout the matrix? How is particle size affecting the drug release? What about the drug load and the surface area? Is the polymer membrane creating, is it the rate limiting step? Is it contributing to the type of mechanism of drug release, or is not playing any role at all? We expect applicants to give us understanding and show to us in their submission, is there best release during the initial release, or there is lag time being followed? Is the drug dissolving before it's, is it going ahead just dissolving in the release media, or there's some diffusion across, like I said, the rate limiting membrane? Hence, we recommend that for the real-time drug release method and for the accelerated short-term drug release method, they follow the same drug release mechanism. And both methods should be discriminating such that it can detect changes when it comes to the CMAs, the CPPs, and the CQAs, particularly including the curing rate, the particle size of the API, that is it picking up or detecting changes in the the drug loading, when there's incomplete sealing, is your method able to detect these changes occurring? I have provided here certain critical quality attribute considerations that we want applicants to keep in mind when testing their final products. I can't stress again enough that the physical appearance tends to be one of the many issues we observe. We've seen many cases of the vaginal systems having discoloration, sometimes it not being fully welded and having incomplete welding, the joints not being smooth enough in comparison to the RLD. In certain cases, we don't see complete studies or data submitted to let us understand that the applicant really have a sense of understanding how the API particle size is affecting the state of the API within the polymer. And dimensions is always critical to show comparativeness to the RLD. And here for polymer impurities, it's really critical that applicants have a great understanding when it comes to some of the byproducts that may be present in residual monomers being contributed by the polymer, extractables and leachables. Please have an understanding of what are the potential leachables that may be contributed by the polymer. In our quality assessment, we recommend applicants follow USP 1663 and use a wide range of solvents for extraction that follow a wide range of polarities. And again, we expect that applicants not only test one batch, but test 3 batches across the stability time points of the drug product. And again, we expect applicants to demonstrate understanding when it comes to their container closure system, that their system, whatever packaging they are proposed is able to protect their final drug product. For biocompatibility, compatibility with condom is a standard test that will enhance prevention of STD. However, when it comes to compatibility with other devices like tampons or lubricants, typically that will follow the ROD labeling. I've already talked about this and I've shared with you some pictures here of samples that we have seen during our quality assessment where the vaginal systems are not completely welded. We have some, when you twist it, the outer sheet is broken, exposing the inner core. These are really critical because they will affect and change the type of mechanism of drug release. In cases where there's flashing at the welded joint, this will impact some irritation in the vaginal mucosa. And bubbles and colorization as well as scratches may affect patient compliance. Some other quality deficiencies we have seen include applicants not demonstrating adequate control strategies when it comes to drug substance impurities, characterization of the polymers. I've already spoken about the residual monomers and the extractable leachables. And again, the mechanical properties, most of the time we see applicants submitting their studies based on only the polymer and not on the final drug product, which again, we stress that is really important. The mechanical studies be performed on the final drug product. And one other area that we see a lot of deficiencies is when the in vitro drug release method is not discriminating enough. I will switch gear and talk briefly on implants. I know we talked about it a lot yesterday. The only thing I'll add to it, it follows the same quality considerations as vaginal systems. The only difference here is the polymers typically used for implants are the EVAs and the PLGAs. We haven't yet seen any unders for next plan on coming, but we have been having quality assessment during pre-unders and where we've had discussions, scientific discussions with applicants who are interested in submitting under application. And I want to stress here that unlike vaginal systems, implants are required per regulations to be Q1, Q2 as an ROD. And here I've provided the list of the typical quality considerations we would expect as fundamentally to be submitted in applications for applicants for implants, including the dimensions, mechanical properties, the impurities, which is a big deal, the applicator dimensions as well, the container closure system, presence of foreign or particulate matter, microbiology sterility. Because it's an injectable, we'll typically expect applicants to demonstrate compliance with USP1 and demonstration of biocompatibility. So in conclusion, vaginal systems and implants are examples of drug device combination products. And Q1, Q2 sameness with respect to the ROD is a critical quality consideration. And applicants are recommended to demonstrate understanding of their CQA, CMA, CPPs, and how the control strategy may impact the quality and performance of their drug products. And when applicants have questions, please go ahead and submit control correspondences. Tell us what you are thinking. If you want scientific discussions, please go ahead and submit pre-under meetings, and we can always discuss and then get to know what your questions are, and then we can help you out. Thank you so much.
Thank you, Cedar. Our next speaker is Ameya Kohojkar. He's a Director of Regulatory Affairs with Pharmathen. Please welcome Ameya.
Thank you, Yogeeta. And thank you, CRCG Group, for having me here. Extremely excited. And good morning, good afternoon, good evening for the audience across the globe. My topic today is industry perspective, challenges in development of PLGA-based injectables. And before I get into the presentation, I would like to speak a little bit about Pharmathen. Pharmathen is a Greece-based CDMO. As of December 6th, there are actually 3 PLGA-based injectables that have been approved. And I'll speak about it as I move along. Pharmathen was the developer of all these 3 PLGA-based injectables. The applications were submitted by Teva, and I'm fortunate enough to have been directly involved in all 3 applications. So with that said, this is the standard disclaimer that I have. The opinions expressed are mine and not necessarily of Pharmathen or its affiliates or my previous employers or their affiliates. So going about and talking about the background for these products, again, there have been some great presentations that were done yesterday. We spoke about background of these polymers. So I'm not going to go into details, but if you can see, there are several PLGA-based injectables that have been approved, right? Until July, there was only one product that was approved, which is generic for Vivitrol. And as I said, as of December 6th, there is a generic that is approved for Risperdal Consta and a generic approved for Sandostatin LAR. So after listening to all the great presentations that happened yesterday, this is a very basic definition of what PLGA is. But PLGA is non-compendial biodegradable copolymer made up of 2 components, lactide and glycolide of varying ratios. There is one topic that actually came in our small group discussion yesterday with respect to the information that is in the GSRS database and what is in the IID database. And if you can see here, there are several PLGAs listed in GSRS database, right? With different ratios of lactide to glycolide. But if we go and compare this with the UNI database, there are probably 6 or 7 listed. So this is one challenge, as I'm thinking of challenges as we move along. Anyhow, these are different polymer structures, that are listed: linear polymer versus glucostar polymer, single versus mixed polymers. Okay. So as a generic applicant, right, what happens? We start with the program, we start reverse engineering the product. Getting a Q1, Q2 formulation is of utmost importance. We want to start submitting a controlled correspondence. So as we know, PLGA polymer is an integral and critical part of the formulation and is one of the release controlling parameters. It is important that we give careful consideration to the PSGs, not only for the product that is being developed, but all other similar products. It was also brought up yesterday that there may be reviews published in different geographies, which may be helpful in developing these products. It is important to be qualitatively and quantitatively similar to PLGA in the RLD, and it does require extensive RLD characterization. Without this information, the agency may not be able to make a Q1, Q2 determination, specifically Q1, which we actually spoke a lot about yesterday. So a controlled correspondence requesting a Q1, Q2 for confirmation could include characteristics such as molecular weight distribution, structure, end cap group, inherent viscosity, glass transition temperature. Again, not an exhaustive list, but at least information that is publicly available that we could include in the Q1, Q2 CC, which would help the agency make that determination. A quick shout out to Dr. Bin. I think this is a slide deck that he had presented in I want to say September of 2019, which sort of gave a good understanding to the industry into what the agency is looking at from for PLJ-based injectable products. All right, so now we have submitted a CC. We have hopefully gotten a confirmation from the agency that it is Q1, Q2. Now we start and dive into the manufacturing process. Obviously, these are parenterals, so there is complexity when it comes to manufacturing these products, right? Drug loading plays a vital role. Simply put, it is the amount of API in the polymer. As we all know, polymers could potentially degrade during manufacturing. Special considerations need to be given to manufacturing conditions. And how do we do that? Establishing appropriate in-process controls to monitor these critical IPCs is important. For doing that, critical process parameters, CPPs, and its synergy with CQAs need to be understood, which means one CQA could impact several CPPs, and one CPP could in turn impact a CQA for the next unit operation. So it is important to understand that synergy, and also important to understand if there is synergy or there is no synergy. It needs to be taken into account as we move along the manufacturing process. It is very important to build a robust and comprehensive quality profile for the drug product. For generic industry, we know we have a QTPP that is established as we start, developing a product. This is something of a live document that needs to be put in place, where you start with a specific quality profile and you start tightening it as you move along the manufacturing process, as you know more about your product. All right, so let's say we have now manufactured a product and you want to test it. I'm not going to go into specific analytical methods. That could probably be a separate presentation in itself. But I do want to talk about the in vitro release method or methods. Most PSGs have a dissolution method listed on them. Information may be available in published review available on FDA's drug approval database. There are additional considerations that need to be taken into account. Two methods need to be developed. One is a real-time method, which evaluates drug release through the intended period of product use, and an accelerated method to achieve complete drug release. Both these methods need to be discriminatory and need to be validated. Due to the complex nature of these polymers, even small changes in excipients or the manufacturing process may or may not lead to significant changes in product release. And why do I say may not? So when we are looking at the discriminative power of an in vitro method, we obviously have to take into account CMAs, CPPs, and CFVs. The FDA recommended range is plus or minus 20%. However, it may not be always possible. And this is also something that came up yesterday, where if there is a temperature condition of, let's say, 5 degrees Celsius, 20% of 5 degrees Celsius is 6 degrees, 7 degrees Celsius. And that may not introduce a meaningful variation in the product. So in such instances, we may have to go and start looking at temperature variations like 10, going from 5 degrees Celsius to 10 degrees Celsius, which is probably like 200% increase. These parameters need to be identified and need to be justified in the dissolution method development report or the discriminative method development report that we put in the ANDA. I now come to another very important milestone. And again, a huge challenge is the BE studies, right? Most of these products need to be studied in patients. What does that do? There is an inherent difficulty in patient recruitment. There are multiple clinical centers that need to be utilized for these studies. Longer duration of studies impact submission timelines, resulting in delayed access to generics. Dosing procedures. And, as we saw yesterday, there are several of these products which are available as combination products. There is an administration device that is provided or a kit that is provided. So what does that do? That, just increases the risk of protocol violations during these studies. So there has been an increased focus on the MI approaches. Commendable work has been done by CRCG and the agency, to tackle this complex issue. That said, I now come to this is, another topic that has sort of, I believe, troubled the industry. And those who are familiar with this topic, they know this is not only a challenge for PLGA-based products, but other dosage forms as well. General expectations are laid down in USP 1663 and 1664. Yeah, obviously missed something on the next point. But anyway, so a comprehensive risk assessment needs to be put together, and that needs to take into account material contact, including material contact time with the manufacturing equipment and the administrative devices that may be available or that may be provided with the product. Current agency expectation is to include method and method validation information for the analytical methods applied for both extractable and leachable assessments. The AET calculation should be based on the correct MDD and a correct SCT for the product. Include toxicological justifications wherever appropriate and possible. Recently, in October, there was a guidance published with respect to quality considerations for topical ophthalmics. It does give a bit of an insight on this topic. The challenges that now remain in front of us, right? So there is lack of common understanding about the information to include from a CMC perspective. As someone who puts ANDAs together, my intention is to provide something to the agency which is easy to review, which is harmonized, which is uniform, and has same information across. There is a guidance that is available for liposomal products, and if something to that effect is put together for maybe a framework for PLJ-based injectables, because there are a couple, that we see do not have approved generics yet. That would be really helpful. There is a lack of understanding regarding requirements for validation of one-time characterization techniques. And again, we touched upon it a little bit yesterday. There are very few polymer suppliers, and they do not necessarily give the right support for the methods that are required to characterize this polymer. So the only option that is left is either you develop it in-house or you go to an external lab. Both of that is time and money consuming, which, as a generic industry, we try to shy away from, obviously. So there needs to be, some sort of a framework, some sort of an understanding on what parameters need to be validated for a method that is put together for characterization. There is a bit of lack of understanding in regarding the nature of microparticles. That is, can there be anything embedded within a microparticle matrix after its formation? Could there be contaminants? Could there be any impurities? Are there any methods that could be used to find that out? Are there any specific sample prep methods that could be put into use that could help us understand that? And the last one, I know this has been spoken about in several forums, is having a framework for post-approval changes for such complex products. And the complexity is this, right? Imagine all these products, as I said, do come with some sort of a combination product, some sort of a device. So now understand there is a device component, there is a polymer component, and if the API is a peptide, then there's a peptide component. How are we going to deal with such changes as we move along? What opportunities has the agency given us? And the pre-ANDA pre-development meeting is a great tool offered by the agency to have early engagement. I've had some great pre-ANDA meetings. It's a good time to come onto the same page with the agency to understand what their expectations are. A new addition through GDUFA 3 post-CRL scientific meetings. Again, we spoke about it a little bit yesterday. And I think, that a lot of people in the industry are taking advantage of this as well. Special mention to MIE pilot program. Again, this is something, as I said earlier, there is a huge push in the right direction towards getting these model integrated approaches available and, to bringing to a level where it could be used as basis of approval. The pilot program started in October. And, again, a shout out to all the GDUFA funded research that has happened, which has helped the applicants to, go through these roadblocks to reach the finish line. Obviously there are some areas that are still being researched on and could be researched on. But it's just a great tool that has been given to the industry at this point. I've just taken the slide deck from AAM. OGD initiated research activities on PLJ-based LAIs in 2013. The first LAI product was approved in July, which is almost after 10 years of starting the research and after 30 years of these products being available initially on the market. That just goes to say, how important the GDUFA funded research is. And that's about it. These are the references. And thank you to all my colleagues at Pharmathen and Teva. Thank you for patiently listening.
Thank you, Ameya. Our next speaker is Dr. William Smith. He is a research scientist with Office of Testing and Research with OPQ and FDA. Please welcome Dr. Smith.
Hello. Okay, awesome. I hope this actually works. Yeah, so I am a research scientist, which means I primarily do internal laboratory research at the FDA. So I am in the Office of Pharmaceutical Quality. Our mission is to ensure quality. That's that slide. Also, this is my opinion and not the FDA's opinion, okay? So what I'm going to be talking about today is phytonadione injectable formulation. So this is really like a 3-piece formulation. So the API is phytonadione or vitamin K. It is an oily substance, so it's immiscible with water. The 2 other primary components, and the one that we're going to talk a little bit about first, is the polyoxyl or the polyethoxylated or PEG fatty acid surfactant that's in there. And that's dispersed in this aqueous phase. And so one interesting thing about this is that the RLD and the RS products here have subtly different ways that the dosage form is categorized here. So either an aqueous colloidal solution or an injectable emulsion. And as you'll see kind of halfway through this, we'll start talking about why that is important. But for our talk today, at the beginning, we'll talk about why, Q1 sameness here for the surfactant is important. And so basically what we're looking at is most of these products have a PEG 35. You can control the ratio of the ethoxylated region here in the castor oil by varying that mole percentage. And so the question is, are all the CAS numbers, so the PEG 30 through 40, qualitatively and quantitatively the same within these formulations? And we were assessing that based on things like critical micelle concentration, particle size, and their formulation behavior. So first of all, hopefully most of you have some idea of what critical micelle concentration is, but basically it's the propensity for a surface active agent or a surfactant to migrate to an interface. And so basically as you incorporate surfactant, you get to the point where the surface is saturated with that surface active agent, then any excess surfactant begins to aggregate into a dispersion, and those are called micelles. And so then this is important because beyond the critical micelle concentration, the propensity for a drug to solubilize or be able to be up or disperse, I guess, into the formulation is more or less proportional to the concentration of micelles. So we did our CMC assessment using surface tension. So this is a drops shape analysis. So this is looking at a change in pressure with respect to the curvature of the droplet, and that is in proportion to the interfacial tension. So as you increase the surfactant concentration, you're decreasing your interfacial tension. And at that kind of critical inflection point, or where the micelles start to form, you get this critical micelle concentration being derived. And so here we can see that the 3 different surfactants that we tested all had reasonably similar CMC values. And I'm just kind of showing both the linear and the log linear versions of this graph here, just so that you can see that it is very easy, if you're not using the log of the concentration or the molarity, to get some kind of erroneous fitting for these. And it's important to use the log because it's fundamental to the actual equations for what's actually happening during the formation of micelles. So stay with the log and the concentration there. And then we took some of these solutions beyond their CMC to measure the micelle particle size. And so we did this both in pure water as well as in a placebo formulation with everything but the vitamin K in there. And you can see equivalent Z average particle sizes from DLS. So we said the CMC and the particle size distribution are not significantly different between these. And we ended up going into manufacture the different strengths with the different surfactants here. And we can see that we have, equivalent pure surfactant, placebo, and phytonadione formulations with more or less equivalent particle size distributions for all three PEG and castor oils. And then this is where it's kind of interesting. You can see these are kind of optically translucent solutions here, which are very similar to what you see in the actual product. So for this first part for Q1, Q2, the PEG and castor oils, similar CMC, similar particle size. They also have similar stability, dilution stability. And so we think they can likely be used interchangeably in the formulation. And I say likely because there are some caveats to this. So this kind of goes into the second question for this product, which is, what is the true dosage form here? And so I know we had an aqueous colloidal solution and an injectable emulsion, and these have slightly different kind of terminologies and cases for them. And really the important thing here again is that those two things are distinctly different because one is a thermodynamically stable system and one is a kinetically stable system. And so this is really evaluated based on the formulation conditions, the type of particles that are formed, and that particle formation. And that's important because the risk associated with solution-like products versus, say, an emulsion are slightly different, where an emulsion also has all of its properties, let's say physicochemical properties, are also closely related to the manufacturing process. And so if we look at what the difference is between, say, a macro emulsion, a nano emulsion, and a micro emulsion, really the distinction here is that we have a thermodynamically stable system and a kinetically stable system. And so as we'll go into here in a second, you can basically formulate these at the exact same composition, but then by using different processing conditions or processing equipment, you can influence the particle size distribution, and that particle size distribution is what is giving you your physicochemical properties. So both like the turbidity here, the optical properties, as well as the drug distribution, where the drug actually is in the product. And so for the thermodynamically stable system, we're usually in that kind of micelle size range, the 10 to 100 nanometers, and it goes up for these kinetically stable systems. And this, of course, the turbidity is a big giveaway here that something's an emulsion versus being an isotropically translucent material. So in general, you can kind of think about this in terms of a phase diagram. In order to make really small droplets of something, you have to overcome an immense interfacial energy. So there's two ways you can do this. You can either increase the surfactant concentration, or you can input a lot of energy to try and reduce the interfacial area, which means you're making more and more smaller particles. And so in general, the microemulsions kind of fall in this compositional region, and emulsions fall in this kind of compositional region. So in order to overcome that kind of immense interfacial energy, usually for nanoemulsions, you're using these really high energy processes. So that's either homogenization or in a lot of cases, microfluidization. And so this is using a high energy input, high shear, to reduce that particle size so that there's sufficient enough surfactant to cover that oil surface and stabilize those particles. This is in contrast to a thermodynamically stable system like a microemulsion, where you can actually make these at really low energy inputs at like ambient temperature with very low shear conditions. And basically what you're looking at is the surfactant and API solubilizing and dispersing into the carrier. And so in general, for a thermodynamically stable system, it should be path independent. So as opposed to the kinetics where your manufacturing process will strongly impact it, if you're looking at these thermodynamically stable systems, you can basically either use a homogenizer or a very slow mixing technique and get the same thing that might take 30 minutes in about 15 seconds. Now, this is only true for one specific kind of direction. And so at that composition, if everything I've showed you so far is adding that surfactant and oil into the aqueous phase and letting it disperse. If you go the other direction, something else happens, and it looks like you can form a nanoemulsion. And this is one of our questions is this an intermediate, or is this a true state for the system? And so it seems like from what we've seen is that if this is a nanoemulsion, it should be susceptible to manufacturing and process changes. And this is important because when you're going through that direction, you're actually going through what's called a phase inversion. And so what happens is you're going from a water in oil composition through this kind of bicontinuous phase, and then you're turning into an oil and in water emulsion. And so what we've figured here is that actually during this compositional inversion, the disturbance of the interfacial area in the bicontinuous phase is susceptible to mechanical shearing and temperature. And so what you end up seeing is that when you go through this manufacturing directionality, you actually get some kind of process variation in particle size. And so that's what we see here. We have the same composition. We can use different shear rates in order to induce different amounts of particle size formation and different amounts of particles ranging from about the 200 to 100 nanometers, as well as our microemulsion we formed using the other directionality. And so if this is truly a microemulsion, you should be able to take two separated phases, and over time, given enough energy, they will form the microemulsion. If you have a nanoemulsion or a true emulsion, just like salad dressing, if you put it in your refrigerator, given enough time, it phase separates. So if we were to take the nanoemulsions we produced, the question is, given enough time and energy, does it split or does it form a microemulsion? And so not to bury the lead too much here, but we take our turbid nanoemulsion, we expose it to some temperature, and here we have a asymmetrical flow field flow fractionation fractogram. These light colors are the concentration detection, and the dark is the light scattering detection. So you can see two particle populations, the micelle portion and the kind of what's actually giving you that turbidity, which is this large particle population in the hundreds of nanometers. And so what's interesting is that it only takes about 3 to 5% concentration of large particles to basically exhibit this much turbidity. So if you were to look at these by batch DLS, you would say these are definitely different, but they actually contain a large amount of that micelle population. But over time, with a little bit of heat, it collapses back to this microemulsion state. And so what I showed you in the beginning is adding your organics into the aqueous. What I just showed you was adding the aqueous into the organic. And there's actually another way that you can formulate this, which is kind of a silly way to do it, but it's to take your surfactant, disperse it in the aqueous phase, and then just put the vitamin K directly in there and to homogenize it. And so what ends up happening there, you get this kind of coarse nanoemulsion, depending on the homogenization speed you are using. And then after 24 hours, oh no, it splits. So it looks like it's doing that classic phase separation behavior. But if you just leave it at ambient conditions for about a month, what ends up happening is it produces this full microemulsion again. So what this is telling us is that at this specific composition that this product is formulated at, the favored state is the microemulsion. So it is a true microemulsion system. And so basically what we were able to show is that, the nanoemulsion will go to a microemulsion giving enough time or temperature. And so these are manufacturing considerations. And what you'll see is that for these different surfactants, they have slightly different physical properties. So specifically the 30 to 50 are very flowable. They're very easy to disperse the oil phase into, and they are syringeable and, have nice flow properties. The 40, on the other hand, is kind of this semi-waxy kind of viscosity to it. They also have slightly different hydrophilic, lipophilic balances. And importantly, all of these are under the same CAS number, just for your benefit here. But so if you are to say, try and make a formulation using any one of these, it's important to think about those kind of manufacturing considerations as far as what temperatures and what mixing directionalities you're using, because you could do this whole process, say, at a slightly elevated temperature and end up always getting a microemulsion out of the system. So in kind of conclusion here, what we showed was that for all three of the PEG and castor oils, they had similar Q1 kind of properties. So CMC, the particle size for the micelles, the placebo, and the in-house formulations. And that when you're using these different surfactants, you have to keep in mind manufacturing and formulation procedures and conditions as far as mixing directionality, temperature, et cetera. And that's especially when you're looking at the kind of flowable ones versus the non-flowable ones. So with that, I think that's my whole spiel here. And I want to just give a special thank to the whole Novel Complex team that worked on this project with me. Thank you.
Thank you, Dr. Smith. Our next speaker is Dr. Rokon Zaman. He is with OTR OPQ FDA. Please welcome Dr. Zaman.
Good morning, everyone. Can you hear me clear? I have a bad reputation of not being heard of. Right. Good morning, everyone. I'm Rokon Zaman from DPQR, Division of Product Quality Research. I'm a staff fellow there under Office of OTR and then OPQ. I will move the slides. All right. So we are from Office of Pharmaceutical Quality. And this is my disclaimer that all the views are here are my views, not the views of FDA. So I'm going to talk today about the release, understanding the release mechanism of integrated systems. So integrated systems are effective and long-term contraceptive options. There are 4 different integrated systems currently available in the US market. All these 4 are from only 2 companies. So although due to the effectiveness of these products, the popularity is going high, but still the cost remains high as well. So one of the ways to reduce the cost is have more generics in the market. But we do have challenges when it comes to developing generics in case of integrated systems. First being the long in vivo application duration of these products. And secondly, the very complex product design. Now, so how we can encourage the development of generic integrated systems. So one way is to develop a accelerated in vitro release testing method that will eventually shorten the in vitro exhaustive long method to establish bioequivalence. And for doing that, we need to understand how the release mechanism of drug from these devices as well. So this is particularly important that because when we accelerate the release from these devices, we don't change the release mechanism itself while accelerating the release. Now, this is a Scylla intertrans systems, as you can see here. And this is a cross-section of this device. So all these device intertrans systems usually have three parts, a T-shaped polyethylene frame, and then this one, and then silicon-based drug reservoir, the inner ring here in the cross-section, and also PDMS membrane that eventually controls the drug release. Now, if we look closely by in this ACM image, this is the part of the drug reservoir. So here you can see the presence of drug particles, solid drug particles. We also examined the membrane itself, but we couldn't find any solid drug particles in the membrane. So from here, what we can say about the mechanism of drug release. So we came up with a hypothesis with the available information that we have that drug may saturate the release controlling membrane initially during storage, and this contributes to the initial drug release upon contact with the release media. Now, based on our hypothesis, this is the concentration time profile. So we hypothesize that the drug release depends on the concentration gradient in the release controlling membrane in this image, this one. And with time, as time passes, our steady state is established when the amount of drug going out from the membrane equals to the amount of drug coming in from the supersaturated drug reservoir. We also hypothesize that the initial release depends on the properties of the release controlling membrane itself. And the properties of the polymeric membrane can be actually explained by Hansen solubility parameter. I'll be talking about that after a bit. Yesterday also during different talks, Dr. Lind and I think Dr. Parks talks, they also explained that how the polymer blockiness and other polymer characterization can be done. But I think HSP, Hansen solubility parameter, can be a simpler way also to characterize polymers. I'll be talking about that in a bit. Now, once we have this hypothesis, next what we did, we checked the permeability of the PDMS membrane in water. So we found that PDMS membrane is impermeable to water or aqueous solvents. So this gives us a very important information that the drug release from these devices is actually dependent on drug partitioning, not by solvent-mediated diffusion. Then we developed real-time in vitro release studies in normal saline at 37 degrees Celsius. We're still continuing this one. This study is more than 800 days now, where we'll be continuing 3 years or maybe a little more. And what is important in this study, if we look closely to the initial hours of this curve, we'll see that there is a phase change from first order to zero order. So the initial first order release actually indicates the release from the saturated membrane, and the transition from first order to zero order indicates the establishment of steady state. So this also supports our initial hypothesis that the drug release starts from a saturated membrane. Now, how long it takes for the drug to saturate the membrane? To answer this question, we actually did several short-term release studies. For these studies, we actually did a release study in real-time condition for 5 hours and then kept the device in storage for a specific period of time. These are the times like 1 day, 2 days, 4 days, 1 week, 2 weeks, and a month. And then we try to model fit the release data. And when we plotted the release rate constant against storage time, we see that after 4 days, the release rate constant became plateaued. So from here, we can conclude that the time it takes for the drug to saturate the membrane is around 4 days. Now, as I've said before, that we used Hansen solubility parameter to determine the interaction of the drug, polymer, and the solvent, and also to determine the possible solvents for accelerated release study. So here we are introducing Hansen solubility parameter for the first time to do this thing, like to use Hansen solubility parameter to determine the possible conditions for accelerated release studies, like solvents and other conditions. So HSP actually predicts the solubility of one material in another based on three forces of the material, like dispersion, dipolar intermolecular force, and hydrogen bond forces. Traditionally, we determine the solubility of one material in another by like dissolves like, but that mostly not that quantitative. So HSP can be a quantitative method to determine the solubility of a material in another. So in HSP, based on the known or experimentally determined solubility of a material in different solvents, we can construct a 3D Hansen sphere in 3D Hansen space, where three coordinates are polar solubility, dispersion, bonding, and hydrogen bond forces. So if we consider these three forces as three coordinates, we can get a solubility sphere in the 3D Hansen space. So here we experimentally determine Hansen solubility sphere for our polymer and drug. So this is the polymer sphere, and this is the drug solubility sphere. And when we overlay these two, we can see that there is significant overlap of the polymer sphere within the drug sphere. So this tells us a very important information that the drug is highly soluble in the polymer itself. And also few different important information that which solvents might be a good candidate for accelerated release studies as well. So we have previously seen that the release is dependent on partitioning of the drug, not solvent-mediated diffusion. So we need a solvent that will not swell the polymer itself, will not go through the polymer itself, but will have a high solubility of the drug. So a solvent should be just outside the polymer sphere, but within the solubility sphere of the drug. So isopropanol can be a good solvent, ethanol can be a good solvent, but THF cannot be a good solvent because it will eventually go through the polymer, and then the release mechanism will change from solvent partitioning to solvent-mediated diffusion. And then we also coined a new term considering Hansen solubility parameter analysis, that is RED area, relative energy difference area. So initially, RED is the ratio of distance of a solvent from the center of a drug or polymer sphere and to the radius of that sphere. And RED area defines the area of from area of a triangle formed by the three spaces, drug, polymer, and the solvent in 3D Hansen space. So we use this idea or new term RED area to analyze already published data. So this is a paper, this is released data from a paper from UConn, Dr. Barges group in UConn. So we analyzed this data and we found that the release rate is inversely proportional to the RED area. So this gives us a very good sense about how useful HSP analysis can be. So if we only know three sets of HSP values of the drug, polymer, and the solvent, we can essentially speculate or predict the drug release behavior of a polymer within a solvent. Now, once we have this understanding about the drug release mechanism, we try to formulate or fabricate in-house intuitive device to do the release studies. So initially, we didn't venture to make the device as it is with maintaining the same geometry, but a simpler geometry that will keep the drug release mechanism same. So here we started this one, the embedded drug release drug reservoir membrane complex, where you can see in this schematic that here is the drug reservoir, and then around it is a PDMS membrane. So this is what it is look like in reality. As you see, that drug reservoir is totally embedded within the PDMS matrix. And the only difference from the device itself is the thickness of the membrane. Here it is around 1 millimeter, while in the device itself, the thickness of the PDMS membrane is 0.3 millimeter. We use this device to do short-term accelerated release studies in the solvents that was predicted by the HSP and also by partition coefficient determination experiments. That data I'm not showing here due to time constraint. Now, the thing is, the prediction of HSP also experimentally proved here that among the three solvents we used, isopropanol showed the fastest release, followed by ethanol and normal saline. That was the prediction from HSP as well. But when we consider this release to the release from the iOS device itself, this release seems really, the amount of drug coming out really smaller compared to the device itself. Why so? This goes back to our initial hypothesis and also here the thickness of the membrane itself. So the thickness is way higher compared to the thickness of the device itself. That's why it's taking more time for the drug to coming out to cross this distance. So membrane thickness is also affecting drug release. So to face this challenge, we started a collaboration with UT Austin group, UT Austin, Dr. Feng's group. I think Dr. Feng is present here too. So they helped us to make membrane of 0.3 millimeter thickness. And using this membrane, we came up with a new membrane reservoir complex, which is exactly the same as the device itself. Like this is the cross section of the device. So here you can see this part, the drug reservoir and the membrane, and this is our fabricated device. So you can see the thickness of the membrane is exactly the same of the thickness of the membrane in the device itself. So, I mean, when it comes to the drug release mechanism, this is exactly the same as the device itself. And then we use this device to do short-term accelerated release studies in different solvents. As you can see, the prediction that we had from HSP analysis also supports our experimental data here. But most importantly, when we compare the drug release from the iOS device itself to the disk that we made in normal saline and normalized by the surface area is exactly the same. So it also proves that the device that fabricated is maintaining the drug release mechanism of the iOS device itself. So, and then also we use THF, and here you can see that the release in THF is really faster compared to others. And this explanation also goes back to the HSP analysis, as I've said that THS will not be a good solvent because this is within the solubility sphere of the drug and polymer both, and that's how it changes the release mechanism of the iOS device. Then we're also doing advanced imaging analysis of the iOS system to see the effect of drug release in the microstructure analysis, microstructure of the iOS system. For this, we have a good collaboration with DGM. And here you can see that there is a significant shrinkage of the volume reduction of the drug reservoir with drug release. So this is partial release. This is an image of the partial release iOS system, and this is intact iOS system. And for the 25% of drug release, there is a 40% of volume reduction. And also we are using AI-assisted image analysis to determine the polymer and drug distribution, and also to see the particle size distribution in the iOS device after drug release. So when we compare the drug particle size distribution in pre-released and partial released scalar iOS devices, we did not see significant difference. So this also eventually supports our initial hypothesis and our understanding about the drug release mechanism, that the lack of significant change in the drug particle size distribution suggests that that the solvent did not penetrate through the membrane. And that's how the particle size distribution remains kind of same. So with that, I'll finally try to say that we have seen that HSP can predict the possible solvents that can be used, and also HSP can be used to characterize polymeric materials like the membrane materials. At the same time, partition coefficient can be used also to predict the possible solvents, but partition coefficient and HSP both depends on medium, process, and material. Now, if we can correlate this medium, process, and material with HSP, we can eventually predict the accelerated release of a drug from a polymer in a solvent based on the real-time release data that we are already generating. So with that, I would like to conclude with my summary that so far we understand that the drug itself, the levonorgestrel, dissolves directly from solid state to PDMS because there is no solvent penetration. And then the release of the drug from the IUS is not mediated by solvent penetration, as the release is essentially mediated by partitioning of the drug. And also the drug transfer between the reservoir to the membrane is another critical part that can be explained by the characteristics of the polymer, and HSP can be helpful in this regard. And also HSP can be a valuable tool to understand and guide IVRT. And finally, by correlating media, process, and materials to HSP and partition coefficient, we can actually predict the release behavior of a drug from a polymer in a solvent. With that, I'd like to thank our division director, Dr. Xiaoming Xu, was my postdoc mentor as well when I started this project, and our team members, our collaborators, and external collaborators. And I work in branch 2, and my supervisor, Dr. Ashtaf, in branch 2, and others as well. With that, thank you all.
I would like to invite the panel for session 4. Hello and welcome back. For our panel, in addition to today's speakers, we have Meenal Chavan. She is with Office of Lifecycle Drug Products within OPQ. Sridhar Desikan, he's a Vice President with Nexus Pharmaceuticals. Siva Vaithiyalingam, he's a Senior Vice President with CIPLA. Dr. Xiaoming Xu, he's a Division Director with OTR. Dr. Rokon Zaman, he's a Staff Fellow with FDA OTR. And Nirav Khatri, he's a Delivery Manager with Dr. Reddy's Laboratories, and I believe he is online. So we'll start with the questions from the audience present here. I can start with the first question. We have.
Fun's got a question. Oh, fun.
No.
Or is it? Oh, there.
So for some of the long-acting implants, they go on for 3 years, 4 years. So to do real-time testing, I mean, you can do it for a few months, but after that, it's unbearable. So I guess, does the agency accept some of the modelings? Because, some of the system follow a very, very mathematically, very clean, models, right? Because, just collect all the real-time data, 3 years. If you got a winter freeze in Texas, you're out of luck. Yeah.
Maybe I'll take on that as a question. So from what you're trying to say is with modeling, how can it help with maybe accelerated release testing? Is that what you're
Well, you can do real time. maybe for a certain period of time, because right, lots of systems are super saturated initially. So real time initial, sometimes you don't know the degree of super saturation. So those real time data is very valuable. But once you beyond a certain period of time, like beyond 3 months, especially after 6 months, modeling can be used to really predict.
Right. The
To
key,
collect the real data over 3 years, just
yeah, that's the primary reason, at FDA laboratory, we started work on the trying to understand how do we accelerate release testing, because if we don't have enough understanding about the mechanism of release, then when you're trying to accelerate even though you want to accelerate the time, but if by doing so you changes the mechanism, then that really doesn't give you the information that you really want to do, which is assurance that the product, when it's during the short shelf life, when it's supposed to release for, not shelf life, but inpatient, 3 years, 1 year, 5 years, however long, that actually it's the, exactly the same condition. So I think understanding is very critical, but how to get the understanding, I think it's a combination of both experimentation and also modeling. And I do agree, with modeling, appropriate incorporation of the modeling with the incorporation of the proper experimentation and understand the whole system, and then you can combine all that and then to devise a simpler experimentation, maybe accelerated condition that give you the same prediction power as you would do over the years of 3-5 years. But we are not there yet, but I think it's trying to get there.
Yeah, because with PLGA based system, so normally some of them doesn't go that long, right? Probably most a year. And then you can use temperature. Temperature can play a huge role, but with all this EVA, PDMS, temperature doesn't drive it. You have to use solvent. Every time you use solvent, there's always a concern about changing release mechanisms and Rokons. It was a nice study. Anyway, I just think, for sure you're going to require real-time distal in just aqueous media, but then real-time distal coupled with modeling, it's a more practical approach. So.
Yeah, but like you said that real time. Yeah, it's working. Working. So you say that how from real time to go to a modeling approach to, that we are working on now. You see that so once we have the whole data for all 3 years, and then we can actually see that what modeling can give us as well. So we're already working with DGM also from that part, like how we can model this drug release from not only from this device, from other devices as well.
Question for Rokon. So thanks for your talk, great stuff. In the intrauterine device you studied, if the release rate in vivo is really dictated by the diffusion rate of the dissolved drug through the membrane layer, what's the actual mechanism for the acceleration using the other solvents? Like, isopropanol was one of the solvents you chose, and it showed, multiple multiples of the release rate. What's the actual mechanism for that? It just I'm having trouble understanding. Anyway, what's the actual mechanism for that?
Yeah. So as the release is actually determined by the partitioning of the drug from the membrane to the release media. Now, the solubility of the drug in aqueous solvent is really low, less than 0.2 microgram per ml. So if you have a solvent that doesn't penetrate through the membrane, but has good solubility of the drug in it, that will help to accelerate that release. That's the approach I think we can have. Xiaoming might want to
Okay.
chime in also.
Just quickly comment on the acceleration, the mechanism, as Rokon has showed, the mechanism through petitioning. So the drug that's in the membrane and then in the soluble form, and then because of the interface between the PDMS membrane and the aqueous medium, the drug is partitioning into the aqueous medium. Now, if you're changing the aqueous medium from 100% aqueous to has some, percentage of organic solvent that have a higher solubility of drug. So this effectively changes petition coefficient or not petition coefficient, but the equilibrium between the membrane and the media. Now you have a higher tendency for the drug to petition from the membrane into this media. It's actually not just a thermodynamic phenomenon petitioning that is driving towards the medium, but actually kinetics also is enhanced because of now it's the medium essentially has a bigger reservoir to allow the drug to go into. So essentially you're driving the drug getting out more and more and even faster. So that is, once the new concentration equilibrium is or steady state is reached, then you reach a new steady state for the drug to dissolve inside the matrix, diffuse into the membrane, and further petition into the media. So by changing the solubility of drug in the release environment can drive all the upstream kind of processes and make it faster.
Gotcha. That makes sense.
So I have a quick point on that. This essentially assumes that the solvent or the media that you're using does not interact with the polymer system, right?
Yes.
So that's very important distinction to make here. So
Yeah, that's my follow-up question. Did you actually look at whether isopropanol swelled the membrane? Did you take measurements?
yeah, I did the experiment, so I better take it. Yeah, we did. It didn't swallow. Like we did, like whether more than 5% swelling is less than 5% swelling. So we cannot even say that whether it's swallowed or not, less than 5%. That's it.
Great, thank you.
Is this? Yeah. So I guess I have a question that's kind of inspired by Cedar's talk about the kind of Q1, Q2, and then Q3. And especially when looking at like all the different PLGA grades where you can have different molecular weights, different molecular weight distributions, TGs. At what point do you begin to start looking that as a compositional variation of the excipient, a Q1, Q2 kind of like structure, or more into the Q3 level of like considerations there? Because I feel like a mechanical, like when you do mechanical testing, mechanical property testing, you get to the point where you're in that Q3 realm of investigating things. However, if you just did the characterization of molecular weight or molecular weight distribution, that could be very easily seen as something under a compositional equivalence of Q1, Q2. So any thoughts on where you begin to draw that line?
So I'm going to repeat the question to make sure I got you right. What you're asking is it okay if you able to demonstrate by testing that your polymer is Q1, Q2, then you wouldn't need the Q3 of the final drug product? Is that what you're asking? So it's not just you demonstrating, like I showed in some of my slides, the process may impact Q3 as well. So it's very essential the final drug product as well demonstrate that comparable Q3 sameness as the RLD, because manufacturing process, maybe stability over the shelf, like may impact it. So just testing the polymer won't be enough. That's why we always want you to show that sameness of the final drug product because of all these additional factors that may impact it. Did I?
Yes, thank you.
I also have a question according the accelerated release testing. Normally, if you accelerate by temperature, it's quite easy to calculate the factor because it's given by a mathematical or physical correlation. If you add organic solvent, how do you get to your acceleration factor, just empirical? And I think it's for each API a different one, or how do you address this challenge?
Question for me, right? Okay, so you can actually define, as I have shown in the presentation, that we coined a new term, RED area. That might be one way to approach it, like how it's interacting with the solvent and drug itself. So that will give you some idea that what solvent you can use and not. And also you can certainly use composition of solvents, like a solvent blend that will change the HSP values of the solvent blend as well. So that might be an approach to select your solvent. But for integrated system that I'm working with is eventually as Xiaoming said before, is at the end solubility of the drug in the solvent. So, and also non-interacting nature of the solvent with the PDMS membrane, I mean the polymer. So combining these three, that should be the way to approach this question, I think.
Just add to what Rokon said. Also, it's not just empirical because when you use temperature changes, they're driven by the Arrhenius equation, kinetics enhancement through the enhanced mobility. But through solvent, you are also changing the mobility by changing the solubilization into the solvent, or diffusion or partitioning. So fundamentally, can be understood, but there is a question of how do you determine it. So that's what Rokon has proposed in this presentation. The concept like solubility parameter, it's a very fundamental measurement about the similarities. It's not like we describe this as a lipophilic or hydrophilic. It's a qualitative, but when you can describe actually a material property using solubility parameters, become much more quantifiable. So using some prediction or modeling, like the HSP with experimentation, combined with experimentation, it actually gave you fundamental kind of an estimate or understanding about the acceleration. So it can be understood, but it does, take extra effort to get there. So we hope, we all can work towards better understanding so that we can get a better assurance of what we are accelerating. We certainly want to speed up the development. We don't want to speed up the development at the cost of, changing the system.
So to continue on that, of course, the solubility parameter is a fundamental chemical property, right? So it's from chemistry. And it will change the concentration gradient. That's the main fundament, basically. That's you are depleting the concentration at the surface, and that's why your solid state diffusion is happening. And probably if you can factorize that, you can reach there, like Rokon very nicely presented. One factor that you, in your presentation, you very nicely showed is this induction time to change the release, right? This is something, solubility parameters is fundamental thermodynamic property. So this is something you cannot predict, the phase where you change, you have the saturation of the film or the shell from the core. Because one factor, of course, even during testing, from the time you begin, you have always this mechanical rejuvenation of the structure of the polymer. When you load that over the time, it will anneal, right? So those properties kinetic. One idea wouldn't be, or the scenario is if you load already, if you have a system where you load the film already with saturated concentration, would that kind of test would, let's say, omit that variation? That's what is your thought on that?
Yeah, I didn't show the data, though. I mentioned that we determined the partition coefficient of drug partitioning from PDMS to the solvents as well. We did this experiment and in the process of even optimizing that too.
Mm-hmm. But I think I provided my message clear, right? Because at the end of the day, it will still not help you to predict the change in the kinetics for the early phase. because that's very stochastic. It's how the polymer will anneal over the testing. Then after you reach the saturation, then your solubility parameter will predict, of course, the later part of the release. But we can talk. So I have one question to Bill. Your presentation, fantastic on the tech castor oil system. And you saw very nicely these different routes of preparation and the colloidal structures found. Have you looked or do you consider that any possible change in the chemical composition of the lipid, or like would that change degradation, or if you have small minor species and difference?
The API or the
the ester itself.
Okay, yeah.
Ester composition.
So I would assume that if it is changing, right, if there is degradation of the excipient, especially during formulation, then that long term, like let's say in the example where you have the nanoemulsion formed and it's going to a microemulsion, so it's going down the step in thermodynamic behavior, that exchange so is about the layability of the surfactant at the surface of the droplet. So the bigger and more well-held that surfactant molecule is to the surface, that interface, the less likely it is to be labile and exchange into the bulk phase. So if you do have some kind of surfactant degradation in terms of like thermal processing, if you're doing this at an elevated temperature, likely that's going to increase the layability of the surfactant, and then it will actually have the propensity to form a microemulsion quicker. Most likely, assuming that you're not getting a big change in the API, and you can also see that through the assay of like loss in potency.
Because the reason I was asking, and we even published this paper, we did some work with a pharmaceutical company. We're looking into MDG, MDG, right? The monodiacylglycerol, which is quite a composition. And this is what is changing over time. And we really tested the MDG of different versions, different batches, different ages. And actually, you really see this is always within the spec, the ester composition, but it does change, and it's a Pickering emulsion. And then since it is changing, you have this change in craft temperature, everything changes.
Yeah. Yes. Very, very interesting. Yes.
Okay. Before we move on to the next question, I would like to give a chance to our online panelist if he has anything to add. Dr. Khatri.
Yeah, probably I missed the chance. There was a question on the solvent selection for the experiment Rokon conducted. So in addition to what Rokon and Dr. Ju added, I hope I'm audible, right?
Yes, we can hear you.
Actually, your audio is not very clear.
Am I clear now?
Yeah.
Yeah. So I was saying that probably I missed the chance. There was a question on the solvent selection for the in vitro release and the aspect of how you choose the solvent. The only point I would like to add on what Dr. Zhu said, that one of the main criteria for the solvent selection, that it should not have any detrimental effect on the formulation what you are checking whether there is a proper release or not. So that is one point I'd like to make. Also, we use sometimes solvents or surfactant for the formulations like liposomes or even microsphere. So whatever component, chemical you are using, it should not have any to release from the formulation when you are using in the in vitro release.
Thank you, Dr. Khatri. Any add-on, or can we move to the next question? Okay, go ahead.
All right, thank you. As a PLGA supplier, I'm wondering, once RLD has been reverse engineered and you understand the excipients that are in the formula, what can we do as a supplier to facilitate demonstrating Q1 and Q2 sameness to take some of the burden off of the CDMOs or pharma companies?
Okay, so as a supplier, basically the first request that we would have is, do you have a DMF for the polymer, right? If you do, The characterization that is required is basically, as I spoke about in my presentation, it's already out there. And if as a polymer supplier, if you can do that, some of these methods can also be leveraged for the finished product. And if we can look at those methods, analyze those methods, use it in our finished product, that would be actually very useful. Does that make sense? Correct. Yep. Actually, I do have something more to add. So when we are looking at polymers, right, we to build a good product profile, we want to test these polymers at their extremes of a specification. And sometimes they may need to be custom synthesized. And I think that would also be a very helpful tool if you have polymers which are at their extremes and at a, at an intermediate, that would also help development of the products.
Okay, we'll have one online question.
Oh,
You want to add? Okay, sure.
Okay. I wanted to add to what he said. We do understand that the supplier has their own quality control for the polymers, but it's also very critical that the burden is on the applicant to demonstrate the effects of the polymer properties on their final drug product. So it's more, the burden is more on the applicant to demonstrate that during their drug product development, as well as in their final specification. Thank you.
So, Cedar, there is a question related to the excipients. So in your presentation, you mentioned that we need 3 lots of API and 3 lots of polymers used in the registration batches. The question is, it's not always possible to get 3 different lots of polymers. In which case, would you accept 2 because 2 is mostly doable?
Putting me on the spot because I don't want to say no, yes, and then set precedence. But I current thinking, we are pretty firm that as much as is possible, 3 lots of the polymer, because it's important to keep in mind that these registration batches you're manufacturing is going to be representative of future batches and supposed to indicate to you some of the quality issues you may see coming up in the future. So I would say currently thinking firmly 3 lots.
Okay, and this is applicable also to the polymers that are very well characterized and they have been around for a long time, right?
Yes, so far as it's classified as a critical inactive ingredient, not such as the polymer, going really great lengths to affect the drug release, and Q1, Q2 as the ROD affecting performance. Yes.
Thank you.
Current thinking.
So just to answer the question on the supplier of PLG and stuff like that, where we get into trouble is when the acceptance criteria for certain attributes. It could be saturation point, it could be the copolymer ratio, it could be the glass transition temperature. So we see many times certain attributes have wider limit. And during development, we are also looking into what happens in the extremes. If you look at the formulation, critical quality attributes, that is function of critical material attributes of the excipients. So the wider the critical material attributes of the excipients, the even more wider the CQA becomes, right? So as you can easily imagine where I'm going, we want a tighter control, so a narrow cut of these critical excipients, functional excipients that comes into play, so we can have a tighter quality attributes in the specification. So part of our development is to define the edges for failure, edges for where we have to take actions. So our request would be most of the time that is cost prohibitive for you because you manufacture in tons and kilograms. whereas our requirement may be few kgs per year. So when we ask for a narrow cut, we have always a tough conversation with the suppliers on the excipient side, especially on the critical excipients. We also see it for non-critical, which we think it is non-critical until at certain point we realize that becomes critical when the limit is wider. I can go as good as microcrystalline cellulose, MCC, right? Such a common excipient. People tend to think, so it's a filler. Yeah, it is a filler. But if the surface area is exponentially high between two lots, that will impact how the dissolution is going to be. So that becomes critical. Thank you.
Just to add from the applicant point of view, if there are very wide ranges, then from the agency, we always ask they have to show at both the ranges that their product is going to qualify. Because if they get, suppose any criteria, like low viscosity, high viscosity. So then during development, they have to give us all the data if they are going with that wide ranges.
So regarding the requirement for 3 different lots of the excipients, I think we spoke about it yesterday briefly. One of the challenges that we have is that, while the test product or the generic applicant is trying to make a product with three different lots, whereas the reference listed drug sometimes could be made with the same lot of material. So you don't really have that much for us to work with, in the sense that in order for us to establish the sameness, it becomes even more challenging because the RLD is pretty consistent, because they use the same polymer for multiple different lots. And if the generic lots are not available for us to compare, then it becomes very difficult for us to establish the sameness. Any thoughts from the industry, from the regulatory perspective, from the FDA on this?
Okay, I can add some comment for that. Oh, here. Okay. Because during yesterday in my talk, I just mentioned about the 3 lots of batches within the same strength of the RLD. Why is that sentence come from? Because we, in agency-wide, we thought about the same thought, because we don't know in the RLDs how much different batches they're using for the final drug product. But as our experience is, if you're using actually, if that is an assumption, is in the RLD has a multiple strengths drug product. If they using normally, they not using they using the different lots of the excipient. And also, it's a generic company also try to do in the same way, in my belief, because when you just make the different strengths, you using the same lot of the excipient for the 3 different strengths, and then you just using the different excipient using for the different strengths. And same as when you're using the 2 or 3 different lots of the drug substance, when you just synthesize of the final drug product, we believe in the same way. And so that's we just consider for the 3 lots of the RLD batch from the same strength. And that's my 2 cents add.
I have a question for Dr. Kohojkar. So you mentioned, right? I mean, right now, I think really significant progress has been made to decide on how to determine or characterize the ILD, so you'll be able to claim the qualitative sameness when comparing the finished product. But once you thoroughly characterize the ILD and knows what is your endpoint, how do you go back to your start point?
Yeah, that's a difficult one, but I think the answer lies in comparability, right? We need to be as comparable to the RLD. So once I determine what the RLD is giving me, we just have to do a trial and error for the most part. Come as close to the RLD as possible. I think that is somewhere of a starting point that we try to aim for. So, as an example, if we do reverse engineering and if I understand there is a linear polymer, then I start looking for polymer suppliers who can give me a linear polymer. I contact 10 different suppliers, if there are 10. We get them, we characterize them, we see what is as close as the RLD. I think this is what one of the discussions was yesterday, right? what is that range where we can say it is Q1 same, but there are still certain minor differences? How can we define, and what are those minor differences? I think that is something that needs to be explored more. I don't know if that makes sense.
Yeah, it does. But I think another challenge or a feedback, and yesterday we may have touched upon during discussion, but not too much in depth, is like how now we know where we're shooting at, right? Then we are trying to see where we can actually further expand the flexibility. For instance, for the starting material, the L2G ratio is to the critical point that you probably can't really vary too much if you don't want to go to really the drawing board, right? So, but then for molecular weight, for instance, that is one parameter depending on the scale of your manufacturing and also the method and the solvent, right? You would choose. You could have different level of degradation during manufacturing, so you could end up with a finished product showing comparable mean molecular weight, but you could actually have some more, more difference comparing the raw material. So I was just wondering like how we could further work to sort of expand that flexibility, or do you think that is something would be beneficial?
I think for this one, I do need to go back and ask my SMEs if that makes sense, but something that I'll take back and, I'll see if we can be in touch and discuss further on this topic.
Okay. You want to add to that?
Just to add on to what Ameya said, it is always a catch-22. So when we do this, like Ameya said, when we do the reverse engineering and figure out what kind of polymer is in the listed drug, then we may come up some other polymers that not essentially Q1. So Q2 is not an issue. Say that is listed in the RLD label, we can find out the amount. So if it is not as Q1 as the agency expects, in our first control correspondence, we get a rejection saying you are not Q1, Q2. But then it becomes big onus on the sponsor to figure the minor differences that we see in Q1 is not really impacting the Q3 or the formulation's quality attributes. That requires an entire gamut of research and development program, and then perhaps a pre-ANDA meeting to get alignment to further continue on this research. Because after we establish Q1 and Q2, then only we can go for further testing and exhibit batches, stability, etc., etc. So this is upfront. We have a lot of work to be done, as opposed to, for example, say a simple injection. There you can relatively easily establish Q1, Q2. But establishing Q1, Q2 sameness in a complex dosage form is really complex. Thank you.
Okay, there are several questions for Dr. Smith, so I would like to move on to.
Yogeeta, may I add to this point, the last question?
Sure.
Yeah. So probably if we are stuck and if we are not finding proper Q1, sometimes exploring more number of lots of RLD may help. And sometimes age of the RLD will also help, because some of the characteristics you cannot see on the initial phase of the age of the product, but some characteristics you can see when it is aged. So probably analyzing different age of the RLD would also help.
Okay, so the next question for Dr. Smith. If phytonadione injection is a self-forming microemulsion, would the characteristics of the product be defined by Q1, Q2? Why FDA still recommends an in vivo approach for establishing BE for this class of products? Why a biowaiver based on Q1, Q2, Q3 is not recommended if it's a thermodynamically stable system?
Good question. It's a recommendation. So if you have alternative methods or alternative approaches that you would like to use or suggest in your development package or submission, we would suggest that you bring those to a pre-development or pre-submission. Do you maybe somebody who knows more regulatory can speak to that?
Yeah, it's a good question. So the risk, I think, depending on, as Billy talked about, the microemulsion versus nanoemulsion or coarse emulsion, depending on the thermodynamics or kinetics, the risks are different. And this risk translates to whether or not manufacturing process play a role or not in the final product characteristics as well as the performance. So if the product is behave like a solution, then the regulatory pathways can go through very similar to a solution, Q1, Q2. So there can be a case to be made that the biowaiver can be bring to the agency and then to be assessed and reevaluated. So it's depending on the risk factor. So a system that's purely governed by the composition, then you can treat it like a solution.
A follow-up question. Since phytonadione is an injection product, does it have to be a nanoemulsion or a microemulsion? I mean, are they interchangeable as long as the formulation is miscible with plasma? What insight does the identity of the dosage form bring to understanding the in vivo performance of this product?
To tell you the truth, I do not know whether or not the either pharmacodynamics or pharmacokinetics between a nanoemulsion like particle size distribution versus like the 15 nanometer micelle or swollen micelle microemulsion would have different disposition. But that's an interesting thing that somebody out there can do and then tell us whether or not it has a big difference.
But as a regulatory pathway, you cannot have two different products. This parental product is by 21 CFR is required to be same dosage form, same composition, right? Not same composition, but same pharmaceutically equivalent. So a nanoemulsion and a micelle are not the same system.
If I could add to the earlier question on the PSG. So coming from the office who is responsible for almost all PSGs, I like to see actually our PSG is work in progress, meaning, we're trying to develop a PSG based on our current understanding. But once we post the PSG, doesn't mean the job is done. But in many cases, especially for complex products, that means we have new projects to be done. So, but we push the PSG out to reflect our current best understanding, to also trying to promote generic companies to consider the product for generic development. And we always look forward to hearing, your feedback as for where our PSG can be improved. And as you have seen from Rokon's and Billy's talks today, we do actually initiate internal and also external research projects to help us better understand, because PSG development is one place. We often identify remaining scientific gaps, and we believe certain alternative novel approach can be done, but we need to do more work to first verify certain things ourselves before we can take that step. So with that being said, everything takes time. Today, hopefully you have learned and feel the understanding of a particular product, you have improved understanding. And based on that, if you believe our current recommendation can have alternative, but it hasn't been done in terms of the revision has been posted, always come to talk to us, propose your alternative based on scientific rationale, and we can get agreement in pre-end meeting discussion or controls.
We are running over time, but I have this one last question quickly for Cedar. What kind of rheological assessment is the agency looking for on the polymer-based systems such as vaginal rings? Is it part of development stage only, or the agency expects industry to have specifications in the final drug product? Once the ring is formed, there is no need for controlling flow characteristics. Is that correct?
So we can't speak to specific, we can make recommendations. You just have to justify whatever method you're using and show how it supports the quality of your polymer. And then final decision will be a review, sure. You want to add?
Yeah, like what she said, it has to support the finished product quality. It will be again case by case basis.
Okay. Since it was a short answer, we'll squeeze in one more question. This is for either Ameya or Cedar.
Can I just a little add my 2 cents? Here.
Yeah.
Okay, for the rheological behavior, we just that is not in the routine test currently our thinking, and that is for the one-time demonstration test. But it's case by case.
Yeah. How do we prove the real-time dissolution profile similarity between RLD and generics containing PLGA? Because F2 comparisons may not be feasible since the drug release happens in stage-wise manner, like lag phase, burst, bulk erosion, diffusion.
So if I understand correctly, the question was, how can we show similarity between the RLD and test product because there are different phases? And if that is...
What kind of test would you use to, for example, for IR, AMR, we have F2 criteria. So for these kind of products, what kind of criteria would you apply?
We have actually applied F2 for the products that we have worked on.
Yeah.
And has worked for us.
Okay.
So.
Okay, so we are over time, so I'm going to conclude the session now, and I think we're going to open for session 5. Thank you, everyone.